أعوذ بالله من الشيطان الرجيم. Bismillah ar-Rahman ar-Rahim. Now, my dear fellows, students, friends, and colleagues, and those who are interested in what I am telling is the interpretation of the blood complete picture of complete blood count CBC. Although it is a routine investigation, should be done in every case of clinical presentation, whether it is anorexia or insomnia or it is what do you call as fall of hair, badness, anything, any presentation, whether it is presented and on the history taking or on the clinical examination, physical findings, you have to advise the CBC and try to understand it. I have already said that there are parts to learn. The first part was the RBC indices. The second part was WBC indices. The third was the platelet indices. The fourth one was the ESR or acute phase indices. And now the last least part is the peripheral blood film. This is section 7. And I am taking peripheral blood film in a very short duration of time. You can understand the importance of this. I am Professor Syed Ali Haza, representing as a visiting professor of medicine, Jinnah Sindh Medical University, Karachi. Now, if you are watching, you have seen previously my slides of the blood CBC. When you see that, the first thing you see is the RBC index, the hemoglobin. And then you see the RBC in cubic millimeter, and then you should note the hematocrit of packed cell volume, hematocrit. If the two parameters are significantly decreased, it is known as anemia. If it is insignificant, although the patient is anemic, as per pathology is concerned, but on the basis of this definition, it will not come to the conclusion, because you have to see the other parameters as well. That is mean corpuscular volume, the mean corpuscular volume, MCSC, mean corpuscular hemoglobin concentration, and mean hemoglobin concentration, MHC. Last three will tell you about how the picture which you have just noted for the sake of the anemia. For example, if you are doing the peripheral blood film and the patient was anemic, then the RBC series on the micrograph, that is in the light microscopy, you can find the different type of pictures. If all are normal, the condition will be described there is the normocytic, means cell size is normal, and it is normochromic, although the patient was labeled previously as anemia, but there is normochromic normocytic. Then you can see also the morphology, morphologically speaking, there are sizes of the different cells are different in the power field. The size are different in different sides of the film micrograph. It is called anisocytosis. there is a picture of microcytes and macrocytes only, means 50% are the microcytes and 50% are the macrocytes. It is called biphasic. And if the shape of the RBC are different in the different sides of the slide, it is called as the poikilocytosis. Poikilocytosis. If there is MCH or color index, I will say the MCH or MCHC will also highly increased. It is called polychromasia. Polychromasia is seen with microcytic anemia, as I have already told you about the condition in which it occurs. So these are the conditions in which you find also the target cells, means the cell are seen under the microscope as a bent, rounded bands, one layer, second layer, third layer, fourth layer, with a specific staining of the methylene blue. So it may be possibility of the target cells. It may be the possibility of the elliptical cells. It may be possibility of the spherocytes. Now these are all, and they are not significant. They are 2 or 1 in number, for example. They are not much. Now this is the presentation that this bone marrow hurry. The RBC are forming in the bone marrow, and because the patient is actually seen or sensed by the bone marrow, the anemia is going on, and the bone marrow responds quickly to form the cells. So the cells are appearing like this: anisocytosis, poikilocytosis, polychromasias, and you will see some target cells and so on, elliptical cells, so on. This is seen usually actually in cases of the anemias. So do not worry about it, because if you are getting this type of the picture and the RBC is concerned, do not confuse it. It is a bone marrow hurry. It is a response of the bone marrow to produce the cells, that is red blood corpuscles. In the same peripheral blood picture, as far as the WBC series are concerned, neutrophils can be detected, their picture can be detected if they are abnormal. For example, they have a large size or they have much granulation, it will be mentioned. And in the same sense, the lymphocytes are described. Typical lymphocytes is seen here. Atypical lymphocyte may be small, may be large, may be more stained. It is atypical type of the lymphocytes. And the banded type of the WBC can be seen also. Monocytes can be seen in the peripheral blood film. The big cell, because it is the precursor, or rather with the progenitor of the lymphocytes. So eosinophil again will be stained with the eosinophilic staining very clearly here. If they are much in number, they will be mentioned. If their shape is different, this will be mentioned also. So RBC series will be described on the peripheral blood film. The WB series will be described on the peripheral blood film, as well as the platelets are concerned. Because the platelets are the smallest cellular elements there are mega platelets you can see. Platelets are seen on the separate actually what is called as the on the slides. Different slides should be made to see exactly the platelets are concerned. If they are aggregated, they are clumped, or they are large in size. Immature cells if are seen in significant amount. For example, the erythroblasts, from the RBC series or reticulocytes in this RBC series, or immature cells from this category from WBC, that is the neurocytes, the neuroblasts, or basoblast, etc., and the eosinophil cells, immature eosinophils, immature monocyte, immature lymphocyte, etc. If they are significantly seen in the blood film, this will be mentioned. It indicates that it is something going wrong. It is not the response of the anemia. It is not the response of the acute inflammation. It is something else is going on. Maybe possibility of the myeloproliferative disorder, and you can catch it from here. At the same time, the platelets aggregation, the platelet sizes, the clumping, etc., will also indicate if there is something going wrong. They are much in number. Thrombocytosis is there. If there is essential thrombocytosis, only clumping and large numbers are seen. But in cases of the essential thrombocytosis, the whole of the peripheral blood film is loaded with the platelets in high amount. The clumping will be seen, the destruction is seen, and megakaryoblast will be seen also along with it. This will indicate that essential thrombocytosis, but possibly of the reactive also, you have to exclude by your other examinations. So the peripheral blood film and any parasite you would see in the blood, hemoparasites, will also be seen here in this peripheral blood film. For example, let's see taking about what is called as the hemoparasites are malarial parasite is the extreme example of it. You can see the schizocytes here, and you can see the gametocytes here. In this fashion, you can detect the peripheral blood film, and you can go for the conclusion that what is the problem is going on. So the peripheral blood film is the important parameter of the blood CBC. If it is not done, you have to mention another investigation. In some lab, they are not doing it properly. So mention it, I want peripheral blood picture. That they will charge for it 200, 300 rupees more. No problem. You should order the peripheral blood picture if it is not included. ESR should also be included. And the new, according to WHO, research work, ESR is parallel with the C-reactive protein also. You have to order the CRP on that separately because they will charge you for it. This is how we actually interpret the CBC. देखिए इसमें जो है ऊपर से नीचे तक आहिस्ता 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 करके देखना है. एकदम अगर आप लगाएंगे, छलांग मारने की कोशिश करेंगे तो आपको कुछ भी हासिल नहीं होगा, क्योंकि यहाँ पे वो खाली आपको फाइंडिंग दे देगा, डायग्नोसिस नहीं बना सकता. ये जो है ना हेमाटोलॉजिस्ट है, वो उसको नहीं पता है पेशेंट को क्या है. आपको तो पता है ना वो प्रेजेंटेशन उसकी क्या है. आप तो बना सकते हैं, ये आपको प्रोविजनली बता सकता है कि एनीमिक पेशेंट है, इस टाइप का एनीमिया है, या इसके अंदर न्यूट्रोफिल ल्यूकोसाइटोसिस हो रहा है, इसमें मैच्योर सेल नज़र आ रहे हैं, इसमें प्लेटलेट बड़े साइज के नज़र आ रहे हैं, वगैरह वगैरह वगैरह. So in this fashion they are giving the conclusion in their report. But this should be parallel with your clinical examination and presentations, or other investigation if needed should be ordered to confirm it. So this is how we interpreted the CBC. Long discussion has been done. I think it is more than 75 minutes total. If you watch these videos in this fashion, session 1, session 2, session 3 to session 7, with a break, take a break, and then see, take a break, and then watch, then you can understand and interpret it easily, CBC in a correct path, in a correct fashion. I hope you can understand it now, and I'm very, very thankful that you have listened to me. I'm highly thankful to you. We will see, inshallah, with another topic of hematology very soon. وَمَا عَلَيْنَا إِلَّا الْبَلَاغُ. Now this is the actually examined for what is this, which I have said just now. Assalamu alaikum wa rahmatullahi wa barakatuh.